Class 11, Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological character to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into their category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characters like structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationship, etc. Classification system for the living organisms have hence undergo several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientific over the time. Table 2.1 Characteristics of the Five Kingdom Classification. R.H. Whittaker 1969 proposed a five kingdom classification. The kingdoms defined by him were named as Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him included cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 give a comparative account of different characteristics of the five kingdoms. The three domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryote kingdom in the third domain and thereby a sixth kingdom classification. You will learn about this system in detail at higher classes. Let us look at the five kingdom classification to understand the issue and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperm under plant. The character that unfurled this whole kingdom was all the organisms including had a cell wall in their cells. This placed together group which widely different in other characteristics. It brought together of the prokaryote bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular organisms. One say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic group green plant, though they also showed a characteristic difference in their wall composition. The fungi had chitin in their walls while the green plant had a cellulosic cell structure. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in the kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plants and both having cell walls, with paramecium and amoeba which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristic and evolutionary relationship Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarity but is also phylogenetic, is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom, respectively, you will deal separately in chapter 3rd and 4th. 2.1 Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical cocci, the rod shaped bacilli, the comma shaped vibrio, and the spirillum is spirala. Figure 2.1. Bacteria of different shapes. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, they are synthesis their own food for inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. They depend on other organisms or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot spring, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows, buffalo, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall, and if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, and have chlorophyll, similar to green plant, and are photosynthetic autotroph. Figure 2.2. The cyanobacteria are unicellular, colonial, and filamentous. Freshwater, marine, and terrestrial algae. The colonies are generally surrounded by gelatinous shield. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen specialized cell called heterocysts. Example: nostoc and anabena. Chemosynthetic autotrophs. Bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the release energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume. Figure 2.2. A filamentous blue-green algae nostoc. Figure 2.3. A dividing bacteria. Roots, etc. Some are pathogens using damage to human beings, crops, farms, animal, and pets. Chlorella, typhoid, tetanus, citrus, canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3. Sometimes under unfavorable condition they produce spore. They also produce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to the another. The mycoplasm are organisms that completely lack a cell wall. They are the smallest living cell known and can survive without oxygen. Many mycoplasma are pathogenic in animal and plants. 2.2 Kingdom Protista. All single celled eukaryotes are placed under protist, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoan under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plant, animal, and fungi. Being eukaryotic, the Protistian cell body contains a well-defined nucleus and the other membrane-bound nucleus. Some of have flagella or cilia. Protist reproduce sexually and asexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group include diatoms and golden algae desmites. They have found in freshwater as well as marine environment. They are microscopic and float passively in water currents planktons. Most of them are photosynthetic in diatoms. The cell walls from two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica, and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposit in their habitat. This accumulation over billions of years, referred to as diatomaceous earth. Being gritty, this soil is used in publishing filtration of oils and syrups. Diatoms are the chief producer in the oceans. 2.2.2 Dinoflagellate. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cell. The cell wall has stiff cellulose plates on the other surface. The most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates very often. Red dinoflagellate example Gonulux undergo such rapid multiplication that they make the sea appear red. Red tide toxins released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in the stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicule, which makes their body flexible. They have two flagella, short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigment of euglenoid are identical to those parents present in higher plants. Example euglenoid figure 2.4b. 2.4.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leave the uglifying organic matter under suitable condition. They form an aggregation called plasmodium, which may grow and spread over several feet during unfavorable conditions. The plasmodium differentiate and forms fruiting bodies bearing spores at their tips. The spores processes through walls. They are extremely resistant and survive for many years even under adverse conditions. The spores are dispersed by air currents. Figure 2.4 A. Dinoflagella, B. Euglena, C. Slime molds, D. Paramecium. 2.2.5 Protozoan. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoan. Amoeboid protozoan. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, false feet as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasite forms cause disease such as sleeping sickness. Example Trypanosoma, ciliated protozoan. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia cause the water laden with the food to be stirred into the gullet. Example: Paramecium figure 2.4d. Protozoans. This include diverse organisms that have an infection spore-like stage in their life cycle. Most of notorious is Plasmodium, malarial parasites which cause malaria, a disease which has a steriging effect on human population. 2.3 Kingdom Fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen a function on a moist bread and rotten fruit. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasite fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause disease in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics. Example: penicillium. Fungi are cosmopolitan, are occur in an air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular fungi and filamentous, their bodies consist of long slender thread like structure called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called Soinocyte hyphae. Other have septae or cross wall in their hyphae. The cell wall of fungi are composed of chitin and polysaccharide. Most fungi are heterotrophic and absorb soluble organic matter from dead substrate and hence are called saprophyte. Those that depend on living plant and animals are called parasites. They can also live as symbionts in association with algae as lichens and with the root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction by spores, called conidia and sporangious spores or zoospores, and the sexual reproduction is by zoospore, acrospores, and basidiospore. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei called karyogamy. Third, meiosis in zygote result in haploid spores. When a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2, and however in other fungi, ascomycetes and basidomycetes, an intervening dikaryotic stage n plus n, two nuclei per cell occur. Such as conditions called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and cell become diploid. The fungi form fruiting bodies in which reduction division occur, leading to the formation of haploid spores. The morphology of the mycelium mode of spore formation and fruiting bodies form the basis of the division of the kingdom into various classes. 2.2.3 Phycomycetes. Members of phycomycetes are found in the aquatic habitat and on the decaying wood. In moist and damp places or obligate parasite on plants. The mycelium is aspetate and coinocytic. Asexual reproduction takes place in zoospore, motile, or by aplanospores, non-motile. These spores are endogenously produced in sporogenium. A zygospore is formed by the fusion of two gametes. These Gametes are similar in morphology, isogamous or dissimilar angiosperm or oogamous. Some common examples are Mycer, figure 2.5a, rhizopus, the bread and mould mentioned earlier, and albugo, the parasitic fungi on mustard. Figure 2.5 fungi A. Mycer, B. Aspergillus, C. Agrarius. 2.3.2 Ascomites, commonly known as sac fungi. The ascomycetes are mostly multicellular, example Penicillium, or rare unicellular, example yeast Saccharomyces. They are saprophytic decomposers, parasites, or coprophilous, growing on dung mycelium. Is branch and separate. The asexual spores are conidia, produced exogenous on the special mycelium called conidiophores. Conidia are on germination produce mycelium. Sexually reproduced are called ascospores, which are produced endogenously in sac-like asci. Singular asci. These are asci are arranged in different types of fruiting bodies called ascocarpi. Carps. Some examples are Aspergillus, figure 2.5b, claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicious. 2.3.3 Basidiomyces. Commonly known forms of Basidiomyces are mushroom bracket fungi or puffballs. They grow in soil, on logs, and tree stumps. In living plant bodies are parasites. Example rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organ is absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is called dikaryotype, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiophores. The basidiospores are exogenously produced on the basidium. Example basidia. The basidia are arranged in fruiting bodies called basidiocarpus. Some common members are agaricus mushroom, figure 2.5c, Astilejo smut, Puccinia rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfect fungi, because only the asexual or vegetative phase of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkage was established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect stage of members of Deuterocyte were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual reproduction known as conidia. The mycelium is separate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of little and help in mineral cycling. Some examples are Alternaria, Trichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae include all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as insectivorous plants and parasites. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and the cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae and include algae, bryophytes, pteridophytes, gymnosperm, and angiosperm. Life cycle of plant has two distinctive phases: the diploid saprophytes and the haploid gametophytic. that alternative with each other. The length of the haploid and diploid phase and where these phases are free living or dependent on others vary among different groups. In different, this phenomena is called alternation of generation. You will study further details of this kingdom in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack cell wall. And they directly or indirectly depend on the plant for food. They digest their food in an internal cavity and store food reserve as glycogen or fat. Their mode of nutrition is a holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults and that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient feature of various phyla and described in chapter 4. 2.6 Virus, prions, and lichens. In the five kingdom classification of Whittaker, there is no mention of lichens and some acellular organisms like virus, prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effect virus can have on us. Even if we do not associate it with our condition, virus did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the virus are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Figure 2.6 A tobacco mosaic virus TMVB bacteriophage. Once they infect a cell, they will take over the machinery of the host to replicate themselves, killing the host. Would you call viruses living or non-living? Virus mean venom or poisonous fluid. Dmitry Ivansky 1892 recognized certain microbes as casual organisms of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they pass through bacteria-proof filters. M.W. Bejernik 1898 demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogenic virus and called the fluid as Contagium vivum fluidum. Infection living virus fluid. W. M. Stanley 1935 showed that the virus could be crystallized and crystals consists largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasite. In addition to protein, virus also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infections. In general, viruses that infect plant have single-stranded RNA and the virus that infect animal have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses are bacteriophages. Virus that infect the bacteria are usually double-stranded DNA virus. Figure 2.6b. The protein coat called capsid made of small subunits called capsomers protect the nucleic acid. These capsomers are arranged in a helical or polyhedral geometric forms. Virus cause disease like mumps, smallpox, herpes, and influenza. AIDS in human is also caused by a virus. In plant, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, drawing, and stunt growth. Viruses. In 1971, Theo Dioner discovered a new infection agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the viroid was low molecular weight. Prions. In modern medicine, certain infection neurological disease were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called Prion, the most notable disease caused by prions are bovine spongiform encephalopathy (BSC), commonly called mad cow disease in cattle, and its analogous variant CR Jacob disease (CJD) in human. Lichens are symbiotic association, mutually useful association between algae and fungi. The algal component is known as phagobionts and fungal component is mycobionts, which are autotrophic and heterotrophic respectively. Algae are prepare food for fungi, and fungi provide shelter and absorb mineral, nutrient and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted air. Summary: biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological character. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic, heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophytes, dinoflagellae, euglenoids, slime mounds, and protozoan. Protists have definite nucleus and other members bound organelles. They reproduce both sexually or asexually. Members of kingdom Fungi show a great diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. The Plantae include all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperms, and angiosperms are included in this group. The cell cycle of plants exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by the asexual mode. Some acellular organisms like virus and viral as well as the lichens are not included in the five kingdom system of classification.